Hello and welcome back. This is the day 2 of digestion, part 2. And all we have left to do is really talk about the chemical digestion, absorption, and then control of digestion. So if we were in class together, we'd probably be eating pizza because that's kind of what I like to do. If we're going to talk about food, I kind of like to eat it. But I'm sorry, we're not. So just sort of imagine you're eating pizza. And so we're going to look at the different types of food. And so think about a pizza where it has carbohydrates, so the crust. So carbohydrates are sugars or starches. So pasta, rice, grains fall into this group. Most of the flour is going to be enriched with iron. So we're talking about iron. Cheese is going to have milk proteins and lipids and calcium. And meat, of course, on a pizza is going to be protein. Also, there's always some sort of fat associated with meat. And then, two, there's iron in meats. And vegetables, the non-digestible fibrous part, their cell walls, that's going to be our cellulose. So it's kind of more fun to be eating and talking about it. I'm sorry we're not together, but that's all right. We're going to have a good time anyway. So vitamins are also included in our food. Vitamins serve as coenzymes. We talked about fat-soluble, all dogs eat kittens, vitamins A, D, E, and K. And we're going to talk about where the water-soluble vitamins, the C's and the B's, are absorbed. Minerals are cofactors in enzymatic reactions. And so this is when we're talking about our electrolytes, so phosphorus, magnesium, sodium, chloride, potassium. So we're just going to kind of hit all of those. Yes, so vitamins and minerals, those are enzyme helpers. That's why we need them in small amounts. They're micronutrients. We'll talk more about them in the nutrition lectures. So we're going to start off with carbohydrates. So we talked about starch. Starch is a large polymer of glucose. We start off our carbohydrate digestion in the oral cavity with salivary amylase. And amylase, it breaks starch into disaccharides called maltose. There are also pancreatic amylases, which do the same thing. So in the duodenum and the jejunum and the ileum, on the brush border is going to be the enzyme maltase. And so ASE endings are always enzymes that catalyze chemical reactions. And so right what's before it is kind of telling you what it's breaking down. So maltase breaks down maltose. And it breaks it down in these simple glucose monomer units, which can then just be through transport brought into the mucosa and then exported out of that mucosal cell, and then it's going to go into the bloodstream for absorption. And remember that that submucosal layer is that vascular layer. That's where absorption occurs. So we do eat other things besides starch. And so disaccharides, so sugars with a glycosidic bond, so just two sugars joined together make up a disaccharide. They're still going to have to be broken down into monosaccharides, and that's going to be done by those brush border enzymes. Once again, they perform the final catabolic step. And so lactose is going to be broken down by the enzyme lactase, and that's broken down into those monomer units, galactose and glucose, which then will be absorbed through the mucosa, then in the capillaries in the submucosa. Sucrose is our table sugar. It's going to be broken down by sucrase through hydrolysis reactions into glucose and fructose. And once again, that's going to be absorbed through the mucosa and then into the capillaries in the submucosa. We're going to talk about processing of carbohydrates. What's going to happen to these sugars, the liver, as it's filtering the blood, all these sugars are going to be converted to glucose. Okay, and it's just kind of reminding you of where these enzymes are. And so sucrase, maltase, lactase, you see they are on that brush border. And that kind of makes sense because we have a lot of motility in the small intestine. And so if we were secreting all of the enzymes into the lumen, we would lose them as they were moved through the food tube through peristalsis. This way, the body saves energy because those enzymes are anchored into the brush border of the small intestine. Okay, and that's just everything I just said, sort of a flow diagram, just to sort of help you if you are a little rusty in your biochemistry. Okay, so let's talk about proteins. So proteins are meats, cheeses, eggs, dairy, fish, legumes, all those things have proteins in them. So there are plant proteins. So we begin our protein digestion in the stomach. And the enzyme that's in the stomach is pepsin. It's going to take those long chains of amino acids and just cut them into shorter chains. And so we have protein, it gets denatured by the hydrochloric acid. Pepsin is going to start to cut those protein chains. And then when the chyme goes into the duodenum, then it's going to be hit with the pancreatic proteases. Pancreatic proteases are then going to cut those longer chains into shorter chains, and then we're going to call them peptides. And we also get some amino acids. And then the brush border enzymes are going to do that final catabolic step, so they're called peptidases. The proteases break down proteins or longer polypeptide chains. Peptides break down those shorter chains into those monomer units, those amino acids that can then be absorbed. And the small intestine is the main site of absorption, really for everything, but for these amino acids. A small amount of dietary protein is lost in the feces. Okay, and that's just kind of showing you another picture of what we just said. Small intestines, that is where we get those pancreatic proteases, so trypsin, chymotrypsin, carboxypeptidases, and they just have different sites. They cut in between different amino acids. I don't care that you know which ones are which. Do be able to recognize if I ask you what is the function of these. These are proteases. They break down protein chains into shorter chains, so into peptides. It's going to be the brush border peptidases that are actually going to cut those shorter peptides into their simple monomer units, amino acids, which will be absorbed through the mucosa and then absorbed in the bloodstream, those capillaries in the submucosa. So this is just showing you that reaction. So amino acids are linked together by peptide bonds, hydrolysis reactions. So with the addition of water, then we get the split and we get those single amino acid monomer units. All of the venous drainage from the intestine goes to the liver, and it's going to be the liver that regulates the distribution of amino acids to the rest of the body. What's weird or unique, I guess I shouldn't say weird, let's not be judgy, about proteins is that we really don't have a protein storage molecule. So these amino acids are going to go to the liver, and so there will be an amino acid pool, but we really don't keep a whole lot of amino acids or proteins just for the sake of storage. They're always going to be turned into something else. So excess will be converted by the magical organ, the liver, and excess could be stored as fat or something else. It'll be used for gluconeogenesis, but we don't really have a long-term protein storage molecule per se. Okay, so last one is fat. So fat digestion and absorption, this is a great little review for us.
Fat digestion begins when chyme, an acidic fluid mixture of gastric juice and partly digested food, passes from the stomach into the initial section of the small intestine, the duodenum.
That's another way to say it.
Despite its name, the inner surface area of the small intestine is enormous. This is due partly to its great length, over 6 meters in humans, but principally to the numerous folds and tiny villi and microvilli that line its inner surface. Digestion requires many specialized enzymes as well as other secretions. The liver synthesizes bile, which aids in digesting lipids. The bile is stored in the gallbladder until it's needed to assist in fat digestion. When the chyme enters the duodenum, a hormonal signal causes the walls of the gallbladder to contract rhythmically, squeezing the bile into the common bile duct and down into the duodenum. The pancreas lies just beneath the stomach. It produces a host of digestive enzymes, including lipases, which are essential in fat digestion. The pancreas also secretes bicarbonate ions, which help to neutralize the pH of the chyme entering from the stomach. This is essential because intestinal enzymes, unlike stomach enzymes, function best at a neutral or slightly alkaline pH. Let's examine the process of fat digestion in greater detail. Fats are hydrophobic molecules, that is, they are not soluble in water. However, most enzymes, lipase included, are water-soluble and require an aqueous medium to function. Left to themselves, fat molecules would aggregate to form large globules that would present only a small surface area where the water-soluble lipases could act. Bile prevents this from happening by emulsifying the fat. The bile molecules have a hydrophobic end, which is absorbed into the fat droplet, and a hydrophilic end, which sticks out. This hydrophilic coating prevents the fat droplets from aggregating. These stabilized tiny droplets of fat are called micelles. Once stabilized in micelles, digestive enzymes from the pancreas can begin their work. Lipase breaks down the fats into fatty acids and monoglycerides. Because these products are lipid-soluble, they can easily pass through the plasma membrane in the intestinal mucosa and enter the epithelial cells. Once inside the cells, the fatty acids and monoglycerides enter the endoplasmic reticulum, where they're resynthesized into triglycerides, combined with cholesterol and phospholipids, and coated with a protein to form chylomicrons. The protein coat serves to make the chylomicrons water-soluble and to facilitate exocytosis. Chylomicrons serve as a shuttle to transport lipids through the body. The chylomicrons leave the mucosal cells via exocytosis and enter lymphatic vessels in the submucosa. From here, they're transported to the thoracic duct where they enter the bloodstream.
Okay, so that was a nice reminder for us that bile emulsifies fats. It's the pancreatic lipases that break them down. So let's take a look at this molecule. The number one dietary fat that we consume are going to be triglycerides. Yes, since we're also eating other cells, we get some phospholipids, we get cholesterol, which we'll talk more about that with nutrition. But look, you can see that it's a really large molecule. And so a triglyceride has a glycerol, a 3-carbon sugar backbone, and then is attached to 3 fatty acids. And so it's large, it's hydrophobic. That is why bile is necessary to increase that surface area so the lipases can get to it. And bile, those bile salts, they're amphipathic. So these little white heads, that is the polar part of that molecule, and then it orients itself so the hydrophobic part of the molecule orients towards the fat and just sort of protects the fat, if you will, from water and makes it into these small micelles. Yep, pancreatic lipases break them down. It's always going to be through the hydrolysis reaction, and you get glycerol, 3 fatty acids, or a monoglyceride and 2 fatty acids. And so this is just showing that process. I'm actually going to click to another one. Fat-soluble vitamins, and I think I said that in the other video, they are going to be absorbed better with other fats. They will get brought in with those micelles. So this picture, it's expressing exactly what that video said. It just shows the fatty acids, the monoglycerides, that they're emulsified with bile. We've got those micelles, they're brought in through the mucosa, through the endoplasmic reticulum. They're actually repackaged as triglycerides again in chylomicrons. And so this is going to be a protein carrier. And then they are through exocytosis, and fats go directly into the lacteals. And lacteals are those lymphatic capillaries. From there, it's going to go, all of that's going to end up going to the cisterna chyli, that thoracic duct, which then empties into the left subclavian vein. Okay, we eat other cells. We eat plants, we eat animal cells, we autodigest our own that have sloughed off of the mucosa. And so the pancreas does make nucleases. They're going to break down DNA, RNA into nucleotides. We don't really talk about it too much. We don't get a lot of calories or anything from this. That's why they're not on a nutritional label. But just know, since we are eating other cells, we can also break down their genetic material. And so we have nucleases, just break things into nucleotides, and then nucleotidases is going to break things down into the base, the sugar, and the phosphate, because we were totally fine with using another organism's chemicals. So when we studied the urinary system, we talked about uric acid. When we use nucleotides, that uric acid is going to be our waste product. It will be excreted in the urine. So let's talk about our veggies, our vitamins, and our minerals. Cellulose is the structural component of plant cell walls. They are glucose units, but they are linked in a way that is, we do not have an enzyme to break those bonds between those glucose units. And so that's going to form the bulk of feces. Vitamins are organic. If they are water soluble, they're going to be absorbed in the small intestine. Minerals are inorganic. They're going to be absorbed throughout the small and also the large intestine. They're basically going to be our electrolytes. Electrolytes and water are also going to be absorbed together in the small and large intestine. You can go back to, I believe it was the second part of the first day, we looked at that slide about that absorption in the large intestine and the small intestine of water and electrolytes. So it just occurs throughout the gastrointestinal tract. There are some exceptions though. So some things are going to be regulated. The amount of iron, the amount of calcium, which does require vitamin D to be absorbed, that is usually going to be absorbed in that proximal part of the small intestine, the duodenum, and is going to be based on body needs, so the body can control the amount of absorption from our diet. So just kind of showing you calcium, iron, folate, which is B9, they're going to be absorbed in the duodenum. We're still going to try to absorb calcium throughout the gastrointestinal tract. Vitamin B12, remember, that one needed intrinsic factor made by those parietal cells in the gastric pits of the stomach, and that absorption is going to occur in the ileum, so that distal part. So throughout the small intestine, we'll be absorbing our vitamins and our minerals. There are some that are special, like I said, iron and calcium and intrinsic factor, the three. Those are the kind of special ones that they're going to be absorbed based on body needs. And vitamin B12 needs intrinsic factor. And when you don't have intrinsic factor, that's when you get pernicious anemia, because B12 is necessary for red blood cell production. So our last topic is going to be control of digestion. And let's think about this. We don't eat the same things all the time, but the body needs to digest it, make sure we absorb everything, but the body also doesn't want to waste resources. Making enzymes is costly. Movement is costly. So there's going to be sort of a three-layer method of controlling digestion. And so this is kind of a messy picture, but I need something for you to look at. So we're going to have long reflexes. So there are things that involve the central nervous system. We are also going to have short reflexes. So this is all neural control. And nervous system control is always sensory input, integration, and then motor output. We're also going to add some hormones at the end that also guide digestion. Let's go ahead and look at the neural control first. Okay, so hey, remember you are cranial nerves. This is the vagus nerve, the wanderer, cranial nerve 10. And so it is going to be the primary nerve that both for sensory input coming in and motor output coming out from the central nervous system, so from the brainstem. And so think about sensory input. What could it be? So throughout the gastrointestinal tract, there's going to be chemoreceptors for pH, for different types of food products. There's going to be osmoreceptors, so they'll know how much solute is in solution. So did you just drink water, or did you drink juice with sugars in it? How concentrated is that? So osmoreceptors. And then every one of those organs has a smooth muscle wall, the muscularis, and inside that wall are going to be stretch receptors. So smooth muscle stretch is how an organ knows that food is there. So is the stomach full? Is it empty? It's going to be based on stretch. Is there food in the duodenum? Can also be based on stretch. Large intestine, based on stretch. So that's kind of sensory input that's going to come in to guide digestion. If you think of motor output, remember motor output is always going to be muscles and glands. We're talking about the gastrointestinal tract. So smooth muscle. And this is going to be either by enteric reflexes, so those short reflexes, or by long reflexes from the central nervous system. And then too, like I said, we'll add the hormones at the end. But think about what can smooth muscles do. One thing for absorption, it can control arterioles. We know that when parasympathetic, we're in resting and digesting because life isn't all that exciting all that time. Most of the blood flow is going to go to the viscera for absorption. Motility, like I said, whenever there's food detected, then that organ is going to increase its motility. So when there's food or when it's around mealtime, which we'll talk about why, the stomach's going to start to churn. When there's stomach emptying, that's movement, so the pyloric sphincter is going to relax and control that emptying. Also, when we start to eat and the body senses that stretch, we get the gastroileal reflex. What happens then is that sort of, hey, we have this new meal coming in small intestine. Why don't you move out all the old stuff? So we get peristalsis and we get a relaxation of that ileocecal sphincter. The small intestine, too, when there's food there and there's stretch, that's the cue for segmentation. Remember, that's a movement that's only in the small intestine. Whenever there's stretch and there's food, that's just going to increase that peristalsis throughout the gastrointestinal tract. The gallbladder is a muscular sac, so that's going to be controlled as well. We can get contractions of the gallbladder. You've got the hepatopancreatic sphincter that is at the end of the common bile duct and also where the pancreatic duct comes in. That's going to relax. That's going to allow bile and the pancreatic juice to go into the small intestines. And of course, we have a whole lot of glands. We've got gastric glands, we've got duodenal glands, the pancreas is a gland, so secretion. So just motor output, what's being controlled is going to be the smooth muscle and the glands. And this picture is just kind of showing you, hey, when there's food in the stomach, that's going to increase motility of the stomach, relaxation of the pyloric sphincter. Okay, so like I said, there's sort of 3 tiers of control. Central nervous system, long reflexes, hypothalamus is going to give us our hunger signal. We'll talk about that next chapter. And then, of course, it's through the vagus nerve, which is the autonomic nervous system. The enteric nervous system, which is going to include the submucosal plexus for secretions and the myenteric plexus for motility. There's a lot of short reflexes, like I just said. When the organ is stretched, it knows, hey, it's time to move, it's time for some secretions. And then there's some hormones. We already talked about gastrin. The small intestine will also release hormones that are going to guide digestion based on what we've eaten. Okay, so looking at this slide, parasympathetic, we kind of covered this already. Rest and digest. And so it's going to be through cranial nerves, so 7, 9, and 10, because we're going to get salivation. We need swallowing, so deglutition. And then, of course, gastric secretions and movement, small intestine secretions and movement. And then, of course, the big mass movement that occurs, so that gastrocolic reflex when we eat as well, that mass movement. So parasympathetic, increased secretions, increased motility, relaxation of sphincters so food can move through the tube, and of course, increased blood flow for absorption. So sympathetic nervous system, this is going to be under the 3 E's, so either excitement, exercise, or an emergency. We don't care if food gets digested then, so it's just the opposite. Decreased secretion, decreased motility. We're going to close out those sphincters. We will just leave that food stuff wherever it is. And of course, decreased blood flow. Blood is going to be flowing to the skeletal muscles, the heart, the lungs, and to the brain. All right, yeah, big take-home message. Stimulate digestion, increase secretions, increase motility. Really not that hard. Okay, so there's 3 phases of control of gastric secretions. So we're going to watch this.
Regulation of stomach secretion is divided into 3 phases: cephalic, gastric, and intestinal. In the cephalic phase, the taste or smell of food, tactile sensation of food in the mouth, or thought of food, sends nervous impulses to the medulla oblongata. These impulses cause parasympathetic neurons via the vagus nerves to stimulate secretion of hydrochloric acid and pepsin in the stomach. The parasympathetic stimulation also results in secretion of gastrin from the lower part of the stomach. This hormone travels through the bloodstream and further stimulates hydrochloric acid and pepsin secretion in the upper and middle parts of the stomach. In the gastric phase of gastric secretion, food has entered and distended the stomach. This distension activates a parasympathetic reflex via the medulla oblongata and also has a direct stimulatory effect on the gastric glands. The result is continued secretion of hydrochloric acid and pepsin. In the intestinal phase of gastric secretion, chyme has entered the duodenum, so gastric secretion is no longer needed. When the chyme contains lipids from the digestion of fats or contains enough hydrochloric acid to bring its pH below 2, gastric secretion is inhibited. The lipids and hydrogen ions inhibit gastric secretion by 3 simultaneous actions. First, they cause impulses to go to the medulla oblongata to decrease parasympathetic stimulation of the gastric glands. Secondly, they set up local reflexes via neurons in the wall of the gut that decrease gastric secretion. Thirdly, they cause the release of 3 local hormones which travel via the circulation to the gastric glands and inhibit their secretion.
So let's talk through those phases. So cephalic, that word means head. So you are totally aware of this phase. Think about it if I were baking cookies or you could smell bread baking. Think of something really tasty, your mouth starts to water. And this is actually a learned reflex. So sort of like Pavlov's dog, ringing the bell. He knew he was going to get fed, so he just started salivating. We are kind of the same way. And so this is going to be from the hypothalamus. So this is a long reflex. Hypothalamus, medulla, parasympathetic fibers, the vagus nerve, also cranial nerve 7 and 9. We're going to get increased salivary secretions. Our stomach's going to start to churn. You know this if you go, oh, I'm hungry, and you hear your stomach growling. You're going to get just increased motility. And then basically what it's doing is it's telling the body, hey, I'm going to eat soon. And so the body's going to gear up and get ready for digestion. So that's the cephalic phase. Once someone actually eats and swallows, so once there's food in the stomach, that begins the gastric phase. And I told you gastric or gastro always means stomach. And so there's stomach stretch, chemical stimuli, arriving food, particularly if it's amino acids, so if it's protein, is going to engage neural and hormonal responses. But we're going to stimulate gastric glands and motility. So this is just kind of showing you through parasympathetic fibers. We're also going to get a release of gastrin from those enteroendocrine cells of the stomach. We're going to get increased stomach hydrochloric acid and more stomach secretions. We're going to increase motility, and we're also going to increase in stomach emptying. That stretch, don't forget about our gastroileal reflex. We're going to get peristalsis and opening of the ileocecal valve. We also have that gastrocolic reflex. That's going to increase colon motility. We'll get the tenia coli. They're going to do their mass movement and move that feces into the rectum, and we may get the call usually around mealtime. Usually it's a little after a meal. Takes a little while for that. But just that is going to be the gastric phase. So gastric always means stomach. So now if we have food coming into the duodenum, so think about it. So here we have duodenum filled with chyme. Sensory and stretch receptors are stimulated. We have sensory impulses conducted to the central nervous system. And then an output is going to be a reduction in parasympathetic activity. So you're like, well, that's weird. Not really if you think about it. Remember all the magic that happens in the duodenum? We've got the bile coming in, the pancreatic juice. We're going to do some segmentation. The whole goal, we're going to get all our digestion or absorption in the small intestine. So that means the stomach's job is kind of done. If it's already releasing the chyme, it's like, okay, slow down, stomach. We need to allow time for the small intestine to do their job. And so it's just kind of a way to tell the stomach, good job, but we want to inhibit your secretions, and that's going to be through the enteric gastric reflex. Since there's food in the small intestine, that's going to increase intestinal secretions and increase that motility in the small intestine. It kind of makes sense. Okay, so some of the hormones, we talked about gastrin already, but one of the hormones that's going to be secreted when this acidic chyme, and especially partially digested proteins and fats, we're going to get this release of secretin from the duodenal mucosa. And another name for secretin, and that's how I always remembered it, it's nature's antacid. So looking down here with me, the acidic chyme enters the duodenum. The intestinal mucosa is going to release secretin and it goes into the bloodstream. The secretin is going to stimulate the pancreas to secrete more bicarbonate ions. Remember, bicarbonate ions are going to be released and neutralize that acidic chyme. The small intestine is not designed to withstand acidity. The stomach has that special thick bicarbonate mucus, and that's why there's not a lot of great absorption from the stomach. But the small intestine doesn't have that because it's designed for absorption, so we really need to neutralize that acidic chyme coming in. It also is going to inhibit the acid secretions of the stomach. And so just kind of remember, secretin is nature's antacid. So just sort of like an antacid you would take at Tums if you had too much acidity in your stomach. Okay, so the second hormone coming from the intestine is CCK. So that stands for cholecystokinin. So chola refers to bile, cyst means sac, and kinin means to move. So this name is telling you what it's going to do. So let's go ahead and look. So chyme with fat enters the duodenum. Cells from the intestinal mucosa secrete the hormone CCK, cholecystokinin. It goes into the bloodstream. It is going to then act on the gallbladder, and it's going to stimulate the muscularis layer, the muscle layers to contract. And so we're going to get contraction of the gallbladder, we're going to get relaxation of the hepatopancreatic sphincter, and then we get bile in the duodenum, which is going to emulsify our fat. It also stimulates the pancreas to make enzyme-rich juice. And so this is another thing that cholecystokinin, it inhibits stomach emptying because if there's fats there, fats take the longest to digest. And so that's one of the reasons that fats remain in the stomach longer is that action of cholecystokinin. CCK also has an effect on how we feel, how satisfied we feel. We feel hungry or satisfied. And so CCK is also a hormone that kind of signals to the brain that feeding is over and that we will not be hungry. So it just suppresses our hunger signal. The third one from the small intestine that we're going to talk about is GIP. When I went to school, it was gastric inhibitory peptide, so that's going to kind of tell you it's going to slow down the stomach. But the new name for it is glucose-dependent insulinotropic peptide. If you come over here, this is kind of a messy diagram. I don't really like it, but I didn't have another one. The big thing I kind of want you to see is down here we had secretin, we had cholecystokinin, and we had GIP, glucose-dependent insulinotropic peptide. What those three are going to do, they're part of that intestinal phase, is they are going to decrease the contractile force and rate of stomach emptying. They're going to slow that down because we need time for digestion to occur. The other thing about GIP, this also signals the pancreas to release insulin. And we release insulin after a meal. So we're in that absorptive state, which we're going to post-absorptive state, which we're going to talk about next chapter. But that is another function of GIP. Big take home, food in the small intestine slows down stomach emptying and gastric secretions and motility. Okay, so you have this in your notes, and don't be daunted by that. We talked about gastrin. We know that gastrin increases acid secretions released by the stomach, so it's going to increase secretions from the stomach. We talked about secretin, which is nature's antacid, so we're going to get more bicarbonate to neutralize that chyme. It also inhibits the stomach. We have cholecystokinin, which says bile sac move, so we're going to get contractions of the gallbladder. Also, we said that's going to inhibit stomach emptying. We also get some more enzyme secretions from the pancreas because CCK is stimulated by fatty acids and broken down proteins. We talked about GIP. It's going to inhibit the stomach as well. We're going to get insulin released and that's going to tell the whole body that there's glucose there and lower the blood glucose levels. The one we didn't talk about, so I'm going to down here, and once again, this name is going to tell you what it does. So soma always means body, somatostatin, and so it's telling the body to stay. So once we have digestion and we've got all this motility, we have all the enzymes there, there kind of needs to be a signal that says, hey, digestion's done, the meal's over. And this is going to be stimulated by breakdown products, so monomer units, so simple sugars, amino acids. We are going to get a release of somatostatin, both from the stomach, mostly we're talking about from the small intestine, and that's going to inhibit secretions basically from everybody. But the stomach, the pancreas, we're going to get reduced blood flow, inhibits bile release, but somatostatin kind of puts the brakes on digestion and says that the job has been done. This one I kind of mentioned, but I want to bring up again when we talked about the migrating motility complex or migrating motor complex. This is what happens when we're fasting. I showed you SpongeBob saying, get that crap out of here. We don't want to leave food sitting or food residue sitting. So Motilin just kind of every hour, hour and a half if we're fasting, just kind of moves things through the tube. And just lastly, just to remind you, it's those enteroendocrine cells in the stomach and also in the small intestine that make these hormones, so CCK, secretin, GIP, somatostatin, and motilin. So entero refers to the digestive tract and those endocrine cells. And that wraps up the digestive system. As always, if you have any questions, you can remind message me. We can do a WebEx if you would like to talk about any of this. I hope you have a great day.